epidemiology was the last thing. Is that right?
Yeah. Yeah.
Okay, so that's kind of where we left off, correct? Is that correct?
Yes.
Okay. All right. All the factors of the disease. This is what makes epidemiology so challenging. So let me give you a little case study here. Okay? There was back in the 1980s, down around the regions of the Four Corners. Where's the Four Corners? Where's the Four Corners of the U.S.? Come on. Geography. New Mexico, Utah, Arizona. Oh boy. Okay. Yeah, let's learn a map here. Okay. This is important because they ask you if you have, if you've traveled outside. I mean, when I go to City of Hope when I check in, the first thing I ask, have you traveled out of the United States? Have you traveled out of California? Why? Because if I come in from Texas, that's a red flag, right? Okay? If I came in from, the Four Corners area, they're going to want to know that. Okay? Why? Because there are diseases that are endemic to this region. What does endemic mean? Always present. Good, good. Okay, so here's your case study. All right, so in the Four Corners region, and I don't know if you've ever been out there, I camp out there a lot because there's nobody. Okay, it's mostly tribal land. Okay, you start to see teenagers, young adults come down with a flu-like symptoms. 24 hours later, they develop what's called adult respiratory distress syndrome and die. Okay? Really severe pneumonia that happens very quickly. Okay? It's within this age group, which is not the age group you usually see get sick, right? Okay, so something's going on. All right? So what do? They send their CDC team out there. They're trying to find out. By the time CDC gets there, they can't find a causative agent yet. They're not able to. They suspect. What would you suspect from what you've learned so far in microbe? Do you think it's viral or bacterial? Okay, some of you say it's bacterial. Why do you think that? What makes you think it's bacterial? Yeah. Okay, it might be something with the soil.
Animal feces.
Okay, might be animal feces. Okay. So, can't find it. We're talking dozens of folks, all right? Dozens of these kids that are dying. Now, we got a problem. We got to be able to find the causative agent and prevent it, right? So what happens? Okay? Whenever you're doing epidemiology, you got to account for everything. Okay? I'm going to give you a hint as to what was going on. It was an El Nino year. What is an El Nino year? What happens to that region of the world during an El Nino year? What happens here? It rains a lot. Okay. What does rain do?
Moistens the soil.
Okay, you got moist soil, so it's not something that's coming up from dry soil. What else happens during El Nino years? What? Well, if you got more rain, what grows?
Microbes.
More plants. Yeah, more plants for sure. Okay, there's more plants, what happens? There's more animals. Okay, all right. So, they keep culturing, can't find anything. Meanwhile, these teenagers are dying. Okay, now, so we've talked about climate, we've talked about geography, we've talked about this. Time of year, it was in the fall. Okay. Culture. All right. What do you know about the Navajo that are down in that area? Does anybody know anything about them? About the tribes? Okay. All right. The epidemiologist that solved this was actually from Utah. He was one of my mentors, and he was really super smart. Super smart. So he went down with the team because they started showing up in southern Utah very sick. And he's like, okay, I'm going through the house and culturing. He found the causative agent in the dustpans and in the vacuums bags. Okay? In the culture, who cleans the house? The teenagers do. It's one of the things they do on Saturday morning. Okay? It's one they do. And so what happened? Well, what he discovered was called hantavirus. Okay? And I know you've seen that before because it's been on a PowerPoint. Okay? Hantavirus. How is it transmitted? It is endemic in the rodent population. It's endemic in the rodent population here in Southern California. It was an El Nino year. Lots of plants, lots of seeds, lots of animals. It started to get cold. Where did they come? Inside the house. They peed and pooped in the house. The most important thing was the pee. They carried the hantavirus. And when the pee dried and you started cleaning it, what happened? Aerosolizes, right? Now it's in the air. The kids that were cleaning inhaled it, boom, they were sick. Okay? Epidemiology is a lot of fun. But look at how many things we have to take into consideration when we're trying to find out the cause of a disease. And it's been fascinating. Have you been following what's going on in the Congo? There was a big shift in the last 24 hours. What did they find? Okay, look it up. Okay, you've got emerging infectious diseases all over right now, okay, in different parts of the world. The one in the Congo is very, very interesting. So, yeah, so if you're going to be taking micro, so, you know, whenever I take a group of students out and we go on a field exercise, or well, it's not an exercise if it's students, it's a field trip, okay, they have to sign hantavirus waivers. Why? Because it's endemic here. Okay, you can see this, and so if it looks like a stripe, black and white stripes, it's probably a zebra kind of thing. But every once in a while, you kind of have to think outside the box, right? Okay, so that's the epidemiology is fascinating. Okay, notifiable disease. They're required by law to collect data. If a disease is communicable, what is it? Host to host. Contagious means extremely what? I can't hear you. Real loud.
Effective.
Yes, extremely effective. Good. Okay. Non-communicable is no host-to-host transmission. Correlation, if one factor is linked to another and is what? Oh, another to have a causal effect. Okay, we got all that. And then cause and effect. One factor is present to cause a measurable change in another factor. Okay? So, just basic definitions that you need to know really well. Okay? All right. Again, here's some tables. Love tables. Do you need to know all of these? No, but this is good reference. Okay? So, for example, if you run into, you've got younger siblings and they're like, oh, I don't know, I think I might have an STI. Okay, and what's one of the most common STIs that can be symptomatic or asymptomatic? Chlamydia. Is that notifiable? Well, you get a look on the chart and see, is it notifiable?
Wait, no, yeah.
Yes, okay, good. Yeah, most of the STIs are notifiable. Okay, so, all right. Let's talk about outbreaks of disease and how we describe them. Okay, incidence is the number of new cases. Prevalence is the total number of infected people at any given time. Morbidity is the number of disease cases. So morbidity, so when you look at the weekly morbidity and mortality report, which they've started publishing again, okay, morbidity tells you how many new cases do you have in the last week. Okay, the mortality rate, mortality means what? A number of deaths during a specific time period, and it's usually expressed in epidemiology is deaths per 100,000 per year. Okay? All right. Good. Yeah.
What was the last P? The entire.
Population. Yeah. How many filled these in before you came in today? Raise your hand. Okay. Okay, good. Good. Makes it easier, doesn't it? Okay. Reporting agencies. We got a number of reporting agencies. If you come down with a communicable disease in the city of Riverside, we've got the health department here. We've got the county health department, Riverside County Department of Public Health. We've got the California Department of Public Health that's up in Sacramento. We've got federal agencies, the Centers for Disease Control and Prevention, and you can look this up, the Morbidity and Mortality Weekly Report, which has started up again, which is wonderful. So we can get a picture of what's going on nationwide. Okay, so this is what's called a heat map. Have you ever seen heat maps before? Yeah, probably. They were very common during COVID to see where things were happening. Okay, this is the number of Lyme disease cases. Okay, yeah, we've got them all over the country, but where is it concentrated? In the Northeast. Okay, and so, you know, that's an area you want to have resources, educational resources, and let the primary care physicians and emergency departments know, hey, we're having a lot of Lyme, okay? And this just kind of shows you the stuff that you can see in the MMWR report, okay? All right, any questions on epidemiology, chapter 13, before we move on? This was a dense chapter, wasn't it? Yeah, totally dense. Okay, so yeah, please make sure terms are critical in that chapter. Okay, understanding concepts like ID50, LD50. Okay, how many went to SI so far this week? Anybody go? Huh?
The first day today.
The first day's today?
Yeah, and tomorrow and Friday.
Oh yeah, Wednesday, Thursday, Friday. How many people are gonna go? Good. Okay, let me tell you a little bit about Nuis. She was the highest scoring student in a couple of semesters in micro. I mean, she is very, very good. So yeah, please go. It will, you know, have you ever had an SI before? Okay, so for those of you who had SIs, they're interactive, they ask questions, you know, that kind of thing. So be prepared to be on your toes while you're there, but you're gonna learn a lot, okay? Cool. Okay, speaking of learning a lot, let's see what you learned back in Bio 1. Get out a piece of paper, okay? Okay, got your blank piece of paper out? No notes, no anything. You're going to see what's in your brain. Okay? In the brain. For 1 minute. You're going to do another 1 minute test. Okay, in this 1 minute test, you are going to write everything that you know about prokaryotic cells. I don't care about eukaryotic cells. I want to know everything you know. Were you supposed to read the chapter before you came in? At least the summaries? Okay, good. Let's go. Here's your 1 minute. Ready, set, go. Okay, I'm going to stop you there because it looks like a lot of you have reached your limit. Okay, let's start in the middle of the classroom. Let's start on this row here. Give us something you came up with in your on your list. What's on your list? No, yours. Huh? Okay, are prokaryotes multicellular? No. Okay, go. Okay, they lack organelles. No, okay, that lacks organelles. Give me something different.
Small capsule size. Unicellular.
Okay, unicellular. Yeah, you got more than that though, right? Okay, do all microbes have a flagella? Not all. Okay, go. What did you come up with? Something we haven't heard.
They're small than eukaryote.
We already saw it. Size is already covered. Give me something else. Okay.
Asexually.
What?
Asexually.
Okay, there's a name for biological when bacteria divide. What is it called? Binary fission. Yeah, just don't say asexual. It's binary fission. Okay, Joseph, give us something. Okay, 4 billion years ago in the fossil record. Okay, give us something. Everything you have. Okay, who's got something that hasn't been mentioned yet? What you got? It's got a what? Okay, you're combining two things here. Yeah, it does have a cell wall, but it also has a plasma membrane. Okay? What'd you get? No. Yeah.
It's bacteria and archaea.
Yeah, those are the two domains, bacteria and archaea. Which domain do we focus on? Bacteria, good, because archaea are the extremophiles. Very good. Anything else? Yeah. Oh no, they have the stones. Yeah. Yeah.
The electron transport chain for cellular respiration occurs between the cell membrane and the cell wall if they have it.
Okay, good. Okay, so the electron transport chain occurs between the cell wall and it's not along the cell membrane, but it's between the cell wall and the cell membrane. Good. Okay, that's what I would expect from Bio 1, not from a group that has read the chapter. Okay? Do you see the difference? When we do this in a little while and we go through what these you've got to, if you could come up with like the locations of the flagella, Lopatricious, Amphitricious, something like that, okay? You don't know it yet, and that's okay, but at least hopefully you all got the basics, the basics. But when you come into class, you should be a little bit above that, right? Okay? So, up your game. You can do this, right? It's all about choices, right? All about choices. How much time are you going to put in? Am I going to make sure I know the chapter before we come in? That kind of stuff. Does that make sense? Okay, yeah, you can do this. You can totally do this. Okay. Let's look at some of these cells because they're so cool. All right. Okay. So, let's talk about some of these cells. What range and size are we talking about?
Microscopic.
No. What units? Units. Micrometers. Good. Micrometers. Okay, and we've got all these mug shots here, and you should recognize this. Which one is this one? Spirochete. What shape is this one? Coccus. What is this one? Bacillus. Okay. That's Bio 1. Those 3 questions from Bio 1. Okay. Now we're going to start getting some more details. Okay. Cell theory. Schwann and Schindler. In 1800s, boy, there's a lot going on. They came up with the idea that all organisms are made of cells. The smallest unit of life is a cell. Virchow comes along and says, cells arise from pre-existing cells. So that leads you to a question. What's your next question? Where did the first cell come from? And he just kind of glossed over that. Okay, but that's okay. All right, so there's some more scientists and cell theory. So we talked about theories in the first thing. Okay, let's talk about the prokaryotic cell and the eukaryotic cell. We're going to start with the prokaryotic cell. We know their size now. Okay? One circular chromosome. Not in a membrane. Very different from the eukaryotes. Histones. No histones. Okay? Did you say histones or ribosomes? Oh, okay, you were right. Sorry. My bad. You said histones and my brain thought ribosomes, and I was like, whoa. Okay, why? Wow. Why don't they have histones? What are histones for? What are they used for? Okay, yeah. In order to get a chromosome, a linear chromosome, you need the histones. Here, you got a circular one, so you don't need histones. I apologize. Yeah. Okay. What size are the ribosomes? What are they? 70S. Yeah, 70S. No organelles. Okay. What magic word that we're going to see over and over again is right here? Peptidoglycan. Say it with me. Peptidoglycan. Hallelujah. All right. Peptidoglycan. Say it, spell it. It's going to be, you're going to see it a lot today. And what is this? How do they reproduce? Binary fission. Okay, binary fission. Good. Okay. Now, I'm going to give you the T on these right here, these prokaryotic cells. These are promiscuous AF. I mean, they just will have genetic exchanges with whatever, whoever, as long. I mean, it's been the key to their survival. I don't recommend it for humans so much, but for bacteria, yeah, they swap like crazy. Okay? What else about prokaryotic cells? They're in the fossil record from 4 million years ago. You think they figured out life? They have. But one of the things that we know from evolution is that it's not the strongest that survive. It's not the smartest. Obviously, I can't find my car every day. So it's always a mystery to me where I put socks, and I have socks on, but and where's my car? And I run into things all the time. Okay? If I was out in the wild, I mean, I survived. I'd be running into the trees. Anyway, it's not it's the creatures that are able to change. And if you're super promiscuous and exchanging genes all the time, what does that give you? It gives you plasticity. So you're able to change. Isn't that cool? Yeah. Scary too if you look at this medically, right? Because if these microbes are always changing, hard to treat. Okay, let's look. Size, which is bigger? Eukaryotic. Okay, paired chromosomes, histones. What size are the ribosomes? ADS. And then polysaccharide cell walls. So there are two kinds of eukaryotic cells that have cell walls. What are the two kinds of cells that have cell walls? Plant cells is one. What's the other one? Fungi. Yeah. The fungi. Why do we care about that in micro? Because if you look at the medications we have to treat fungal infections, a really easy target is that cell wall in fungus. And what is that cell wall made of? Chitin. Good. Yeah. Don't say chitin. Please don't say that. Okay? And we divide using mitosis, so we have a mitotic spindle. Okay, cool beans. All right. A bunch of mug shots of all these bacterial shapes. Okay. Ooh. All right, let's talk about some famous ones on here. What famous one stands out to you right now? You look at it and you're like, oh yeah, you're famous. Where? Yeah, there's Strepto. Vibrio. Yeah, they're funky. Okay, what about this one? Okay, know anybody who's got an ulcer? Okay, 75 to 80% of the ulcers are caused by this one. It's H. pylori, Heliobacter pylori. Okay, so if you suspect an ulcer, you get a breath test to find out if that's it. Now, there's some that are not caused by Heliobacter. Okay, but yeah, so you can kind of get a. Oh, here's mycobacteria. Oh, that's been a problem this winter. Okay, so let's talk about morphology. They come in pairs, they come in clusters, they come in chains, okay? So, you know, all the cocci here. These look like a bunch of sausages from the New York delis. You go into a deli in New York and get sausages and they come and they're on a, yeah, good, tasty, really bad for you. Okay, and here's my personal favorite, Stella. Yeah, why do you think they call it Stella? It's a star. I've only had this cultured once in all my years of teaching by, you know, when you students, you know, do their fingers and that. I've only had that once, okay? And I've never had the rectangular bacteria that looks like a bunch of, I think they look like those things of chocolate that you break off. Yeah.
They look like bars of soap.
Bars of soap, yeah. Never seen that one cultured, but I have seen one Stella in 30 years. That's about it. Okay, so let's look at this. Okay? All right, if you didn't look at anything else coming in, hopefully you looked at this. Okay, this slide before you came in. Okay? And there's some things that are familiar here, right? Okay, we see bacterial chromosome. Okay, that's a bio one thing, right? Bacterial chromosome. And where does it hang out? It hangs out in this area called what? The nucleoid. Okay. We've seen this. What is this? Flagella. And what does flagella do? It's for motility. And you've got 360-degree rotation. Acts like a whip. Okay. Don't confuse it with cilia. What is cilia? They were like windshield wipers, okay? This gives you a lot more power. All right, cell wall, okay? What's the cell wall made of? Peptidoglycan, okay. We see a cell membrane. What's that made of? Phospholipids, okay? And then ribosomes. What are ribosomes used for? Protein synthesis. Okay, can you find on here what is new? Let's start with here. Glycocalyx. Kind of introduced it a chapter ago about a glycocalyx. All right. This is a layer of molecules that are external to the cell wall. Okay, and it's used for adhesion primarily, and it serves as receptor functions. What is it made of? Well, the name tells you. What does glyco mean? Sugar, yeah. So it's primarily sugar, and when you see sugars, they usually are adhesive. Okay, so that's a new structure that's new to micro. Okay, all right. What is this one right here, Phyllis? What is this? What does it do? It transfers DNA. It's like the bacterial penis, right? Okay, this is going to transfer DNA. Sometimes it transfers other things as well, but you can also they also can be used for adhesion. You're going to see examples of both. Okay, mesosome. This is an extension of the cell membrane. Look at this right here. Do you see it? That's a mesosome. It's an extension of the cell membrane that folds into the cytoplasm and increases surface area. What do you think is going to happen along there? Cell respiration. And specifically, what happens on a membrane in cell respiration? The electron transport chain. Okay, you learned that in Bio 1 too, and anatomy as well. So, yeah. So when you've got, remember in the ETC, you've got embedded proteins in here. And so this is where it happens. That's where the magic happens, is in those mesosomes. All right? Okay, fimbriae. We've seen fimbriae before. What are they used for? This is all attachment, right? Okay, adhesion. Inclusions or granules. This is new. A lot of the eukaryotic cells don't have them. Plant cells will, but eukaryotic cells will not have inclusions, okay? Could be fat, phosphate, iron, okay? Yeah, you can have magnetic bacteria, okay? And then the inclusion includes iron, okay? So, those are the new things from that. look at your list that you just did in the 1-minute test, and without looking, add in all you can right now. Let me turn on the lights. Give you a minute to add what we just talked about. Okay, let's see what you got. Did you get these? Okay. Did you get these? Whoa, where did it go? There we go. Okay, did you have glycocalyx pili, fimbriae, inclusion granule? There's only 4 new things that you should have that we just talked about. Okay? Does that make sense? Okay, does it help to do this and then review it real quick so it gets in there? Okay, because I only see you 3 hours a week, and that's not enough time for repetition at all. So try and do it as much as possible. Okay, let's talk about the glycocalyx. Okay? All right. What is this glycocalyx made of? Okay, we know this one. Glyco is what? Sugar and protein. It can be made of protein too, okay? There's 2 types. Slime layer, which looks exactly like it sounds, right? Slimy, disorganized, not tightly in, it kind of extends out. That's a slime layer. And then our other flavor that it comes in is a capsule, which is more organized, is closer into the microbe, okay? Now, what does it do? It functions as, you see glyco, what should you think? Sugar, sticky. So attachment, protection, because if it has a capsule, then it can't be phagocytized. And receptors, especially if it's got protein, okay? Proteins make a lot of the receptors, okay? So, what does this do? Well, a couple of cool things here. All right, yeah. So let's talk about biofilms. Biofilms are groups of bacteria that work together, and when they are all working together, it's called quorum sensing. It's pretty interesting. Take the time to read about quorum sensing, okay? Because it's really strange how they look so simple, but they're able to communicate and work together as a team. And what they do is once they all attach, once you get your pioneers there, then they divide and you get more and more, and it can recruit other molecules, and these cells start secreting carbohydrates and proteins and lipids, and they make their really complex community there. Okay? This is what it looks like. What kind of electron microscopy is this? What does that look like? That's scanning because you can see the surface. And then here you can, this is a transmission where you can see the capsule that's there. Okay? Once you have a biofilm established, biologically, in your patient, in yourself, they're hard to get rid of. Why? Because you're destroying a whole community. It's not like you're just trying to kill a few cells. They've got chemicals and that. You say, okay, well, I'll just treat with antibiotics. No, because the antibiotics can't penetrate here. This is why you brush your teeth, right? This is why you brush your teeth, because every morning you get a biofilm. Do you have a biofilm right now? Probably. Okay? Doesn't take long. How these divide every 20 minutes, right? So it doesn't take long. Okay? So what do you have to do? You have to mechanically. Do you clean your toilet? Yes, because if you get a ring, especially with how many total dissolved solutes we have in the water here, okay, if you get a ring, it's a biofilm. You can put chemicals in there, it won't do it. What have you got to do? Scrub. Scrub, scrub, scrub. Okay? So, let's talk about some of these. The CDC estimates that 80% of chronic human bacterial infections are from biofilms. Do you think this is going to be important? Yeah. Dental plaques, infectious kidney stones, endocarditis, catheters, implants, and cystic fibrosis. Why is that important? What is cystic fibrosis? What? What is it? Tell me. What is it? Yeah, you get a lot of mucus. Okay, so in cystic fibrosis, you have a genetic mutation that changes your calcium channels. And so the chloride. Chloride is always open, so it causes an electrolyte imbalance. What happens? Mucus gets really sticky in your lungs and in your gastrointestinal tract. When that happens, okay, what is mucus mostly? It's a bunch of carbohydrates, right? A bunch of carbohydrates. So, yeah, you got carbohydrates, microbes are going to take advantage of it. Okay? You want to be a gazillionaire? Figure out how to prevent biofilms from forming on catheters. I believe it can be done. Okay, why? I had a student back in 2011, didn't take micro, took Bio 1, okay? Took Bio 1. We had a experience that we offered to some students that they went over to UCR Engineering and Environmental Engineering, okay? That's one of my hobbies, is doing a lot of work with environmental engineering, because these bacteria are so important and we need to know how they move in the soil and all sorts of things. So he went over there to do an internship. What he did is he started looking at nanoparticles. What are nanoparticles? You heard of them? You're eating them all the time. They're in toothpaste. They're in white frosting. They're in anything that makes food shiny and bright. Those are food grade nanoparticles. If you have dry fit clothing, if you have makeup, okay, you're putting nanoparticles all over your face, in your laundry, everything. Do they get filtered out by municipal water system treatment facilities? No, too expensive, way too expensive. Do they enter your cells? Yeah, the data says, yeah, they do. Okay, so now let's look at what he did. He developed a paint additive that you put into marine paints that prevents biofouling, and he stopped it at this first step, so that because of those nanoparticles, they were infused with some metals, it stopped biofouling. The Navy was thrilled. 40% of their fuel cost is to carry around all the things on the hull of that ship, right? It's all the extra things, because you lay down some bacteria, you add some barnacles to it, and suddenly that ship is heavier. So it increases your fuel cost by 40% on average. Okay, so he invented a paint that prevents that. Okay, is he a gazillionaire? No, unfortunately, when you work for a university and you come up with an invention, they get the patent and they get the money. So he doesn't. Yeah, is it fair? No, but that's what you sign up for, so yeah.
But you can't do it on yourself. You need all this equipment.
Yeah, you need all the equipment and everything. So yeah, he is a PhD in chemical engineering in Connecticut. That's where he is now with his family of 5. But yeah, he was Bio 1. Bio 1, that's it. So when I say come up with a way to prevent biofilms on catheters, I'm sure one of my students will come up with it. I know he will, okay? Because you're gonna be thinking differently than what other people think. And that's really all you need is in order to do something new. Okay, let's talk about flagella. These flagella are pretty cool, okay? If you notice, there's a difference between gram-negative and gram-positive flagella, okay? All right, so flagella are long, thin helical structures composed of a protein called what? Flagellin. Clever, huh? Easy to remember. Flagellin. Yes, okay. And you notice it's got a hook. Okay, you can see the hooks in both the gram-negative and the gram-positive, and a filament, and it's that filament that's a long whip. Okay, so what you're doing, you're seeing how it attaches. Here you've got the gram-positive cell wall. You've got one of these rings, and in gram-negatives, you've got 2 of those rings. Okay, all right. So far, so good. Okay, let's see. You've got basal bodies. You see the basal body here, and it functions in motility, rotates 360. And yeah, maybe have 1, maybe has 2, maybe has none. Okay, so you're not always going to see flagella. Okay, now we use the proteins that are in the flagella have what are called H antigens. So that means that each species, okay, so if we look at all E. coli, so E. coli has flagella. So if we look at the E. coli, we can tell the different serovars of E. coli, okay, based on their H antigens, okay, what proteins, okay, what different kinds of proteins are part of that flagella, okay. Here's a good example. There's lots of E. coli strains, lots of them, lots of serovars. This is the one that can be deadly. E. coli 0157H7. That tells you immediately it's got flagella. It's got an H antigen, and it tells you it's number 7. So for microbes that have flagella on it, oftentimes we use those H antigens to help designate the serovar. Okay? Is that cool? All right. So this is one of those great things you could have filled in. What word belongs up here? Monotricious. Mono meaning 1. Which in here is monotricious? B. Okay. Lophotricious. Amphitricious. Paratricious. Okay. And if you have flagella all over, like this, you're slower. You might have flagella like D that's at both ends. Or you might have tufts of flagella on one end, like in A, okay? Do you think you need to know these?
Yes.
You do? Okay, cool. All right, let's talk about how these things move. They use what is called taxis, okay? So there are 2 things that attract microbes, chemicals and light, okay? They also repel. Chemicals and light repel. So, and if you're moving towards or away from a stimuli, if it's a chemical, it's chemotaxis. Okay, can you see the light? Chemo. And then phototaxis is what? Light. So these microbes can move towards the light or away from the light. They're photosynthetic. Which way are they going to go? Towards the light. Are they going to be medically important if they're photosynthetic? No, because what do we look at? We look at the ones that are going to attack you. Okay, now bacteria like this, oh, it's so cute, okay, rotates on flagella to run and tumble. How in the world does that work? Okay, yeah, so I will do an interpretive dance of run and tumble. Okay, all right, these are going to be my flagella. Okay, so if I'm running, all the flagella are here, right? And then when I want to slow down, whee! then you tumble. Woo, I'm not gonna do that, this is that. Okay, I'm old. All right, and then when you run, the flagella's all up in here, and then when it wants to slow down, what does it do? All the flagella, whee! Okay, there you go. That's the only dancing we're gonna do. Don't do it in the club, all right? And if you do, I want pictures. I'll pay for that. Yeah. So, okay, so those are run and tumble. All right, now, this makes sense. Think about E. coli that has flagella. Is it easy to move to the vagina? Easy to move to the urethra? Yes. Why? Because it's running and tumbling. Okay? It's going to do that. And if it's damp, it does it even easier, right? Okay. So if you've got, infectious wounds that are weeping, okay, think about run and tumble. Okay? Run and tumble. Okay. These are really beautiful. These are axial filaments. Okay? They're going to be found in spirochetes. They're endoflagella. Endo means what? Inside. Inside, okay? So they're between the cell wall and the outer sheath. They're anchored at one end, and they cause a corkscrew motion. Makes sense. What do spirochetes do? Burrow into tissues, right? Okay, so and they can do that because of axial filaments. Yeah, I know, it's just so much. All right, my second favorite F word, fimbriae. What are fimbriae used for? Yep, they're sticky, attachment. Okay? Boy, E. coli, it's a beast, man. Not only does it have, a lot of them have flagella, but they got fimbriae. Where is it going to stick to? Like inside the, where is E. coli? You're going to sample for E. coli. Where are you going to sample? What part of your anatomy are you going to sample for E. coli? Okay, which intestine? Large, yeah. Large intestine. Good. Okay. All right. Here's another one. What does this microbe cause? Gonorrhea. Yep, another love bump. Okay, why? Look at how the fimbriae. Where is it going to stay? Inside your reproductive tract, in your vagina, okay, in the cervix, in your urethra if you're male, okay? It sticks inside there because of the fimbriae. You can see this. This is gorgeous. Look at this. This is the intestinal. Here's a microvilli, and here's E. coli sitting right there, all fimbriaed out. Okay. So, all right. Okay, time for a little bit of Barry White. Okay, transfer. What is this? Yeah, and what does it do? Transfers DNA. You're never going to forget that, right? Okay, there's two kinds. There's attachment pili, and then there's conjugation pili. Conjugation means those are the pili that are exchanging DNA. Okay, all right. And here you can see this one is just exchanging viruses. I mean, they are, yeah. Okay, when we do microbial genetics, it will blow your mind. It's like, really? You guys do all that? Wow. Okay. Now, let's get to the cell wall. Here's the magic word of the day. Peptidoglycan. Okay? So, it's glycan, which means what? Sugar, with cross-links of protein. Okay? So, if we look here, we've got NAG and NAM. I don't care that you remember acetylglucosamine. Acetylglucosamine. NAG is much easier, isn't it? So if you can remember NAG and NAM, okay, so this is how it's put together. It's kind of put together like you would put together a really good fabric or fence or something. Here's the units of NAG and NAM, cross-linked by polypeptides. So that's what holds it together. It's pretty strong, actually. Okay? But it also, because of the way these are, the proteins are, it gives it a little bit of flexibility, which is what you need. Yeah? What does a D stand for? It is disaccharide. What is a disaccharide? Yeah. Okay. Now, let's look at this. Gram positive. Okay, gram negative. You did these stains, right? Did you do these last week?
Yeah.
How did they turn out? They turn out pretty good? No? Oh, did you do it this week? You do it today? Oh, this is so exciting. Okay, this is really exciting to see this because gram positive versus gram negative. Whenever you get a microbe clinically, this is the first thing they'll ask. Is it gram positive or gram negative? Because that tells you what antibiotics you can use. Which one can you use penicillins against? Gram positive. Okay? If you're trying to treat a gram negative infection, you have to have another target, like ribosomes. Oh, or topoisomerases, gyrases, or something that interferes here. Now here's the key here. These, for the gram positive, okay, may I ask a favor? Unless it's an emergency, please don't get up. Okay? Taking a phone call, whatever, going out, I don't know, but unless it's emergency, please don't. Because what happens when everybody looks back there? Okay? You're adults, 90 minutes. If it's an emergency, I can understand that. Or if you've got a medical condition, please let me know. But just getting up because you need to stretch your legs or whatever, and I'm not saying that's why you got up, okay? But yeah, don't do that, please, okay? So when you've got gram positive here, you give a penicillin. If the cell wall is already intact, that penicillin won't do anything. When does penicillin have its greatest effect? During binary fission. And so you have to take it long enough so that those cells will go through binary fission. Because what does penicillin do? It interrupts that matrix formation that we just saw. Okay? So we go here, here's gram positive, this big peptidoglycan layer with teichoic acid and lipoteichoic acid in there. You've got the cell membrane that's here. Okay, there's your gram positives. And we'll see some colors in just a minute. Gram negatives, you've got a little bit of peptidoglycan. Just a little bit. Okay? But what have you got? You've got an additional layer up here. And you notice you've got lipopolysaccharides, phospholipids, porins, which are pores, protein, transmembrane proteins that go through there. And you've got this really interesting thing right here called the periplasmic space. There's a lot of interesting chemistry when we do metabolism that happens in that periplasmic space. Okay? All right. Okay, let's focus on the gram positive cell wall. Okay. The gram positive cell wall. What's our magic word for today? Peptidoglycan with teichoic and lipoteichoic acid. Okay. When you do gram stain, you use crystal violet, and it stains purple, and it's retained by the microbe because of all that peptidoglycan. Okay. All right. Here's another table that has lots of interesting information. Do you have to reproduce it or know everything? No, but it's there. Okay. Here's our gram negative wall. Okay, our gram negative wall shows an outer membrane. Okay, that's lipopolysaccharide. Okay, and we have lipid A. We talked about lipid A last time. What does lipid A do? What kind of toxin is it? Oh, come on, this was a while ago. What kind of toxin is it? It's an endotoxin. Yes. Okay? And it can be released during infections. So you've got porins, you've got proteins along here. You got a thin layer of peptidoglycan with no teichoic acid. Large periplasmic space for metabolism. And you've got this lipopolysaccharide layer that's got O polysaccharides and lipid A. Lipid A is the endotoxin. Okay, so what happens when that dies? That is released. Okay, can cause sepsis. We talked about this last time. All right. What color does it stain? Okay, color is pink. So what happens is the crystal violet doesn't stick around, and then you counterstain with safranin, and then you get that lovely pink color. So you're going to be doing that. That's going to be fun. Okay. Here's another table of medically important gram names. Okay. What color are the positives? Purple. And what color are the negatives? Pink. Good. Okay. So you're going to get lots of practice. Okay. Lots of practice in lab. All right. So, yeah, got a few more minutes. Let's see if we can get through this. Okay. So, yeah, penicillinase. Okay, so when we're trying to kill bacteria, okay, we use penicillin. That's the most famous, which inhibits the peptide bridges and peptidoglycan. Okay, what is the L? What? Lysozyme. Yeah, okay. And then for M, what is it? This is a mycoplasma. Okay? All right, mycoplasma. When you digest that cell wall, you're left with just a cell membrane, and it results in spheroplasts or protoplasts. Okay? That cell wall has prevented that microbe from bursting. When you digest the cell wall and you're left wallless, now this is particularly sensitive to lysis, and this is what it looks like when it does. Okay, so that's the beauty of penicillin and these wall destroying agents, is then you can lyse the cell. Okay, is that cool? All right, so let's talk about atypical cell walls. Atypical walls, okay? Mycobacterium. The Mycobacterium species are gram-positive with, what, 60% long-chain fatty acids, okay? 50% or 60% long-chain fatty acids. You can't use gram stain on Mycobacterium. And if they're doing a Mycobacterium, they're often looking for TB, tuberculosis, okay? So what they do is you do an acid-fast stain for these cells, okay? Because they don't have a thick peptidoglycan wall, so they're not gram-positive, but they're not gram-negative either. They've got a different membrane composition here, so that's why you have to do acid-fast staining, okay? I don't think we have you do acid-fast anymore because it's a very difficult stain to do. I don't think so. Is it on your schedule for today? I didn't see it, so yeah. So you do gram-positive, gram-negative, you do endospores, but I don't think we don't do acid-fast anymore, okay? Mycoplasma species, they have no cell wall. Archaea, some of them lack cell walls, but they have permeable peptidoglycans, so these are really strange. And so, you know, is it critical that you learn how to do this stain? Not really. But one of the things about the walls that you have to remember, and especially if you're looking at TB, is that these walls are hard to penetrate. Antimicrobials do not penetrate these walls. How long are you on isoniazid for tuberculosis? Any idea?
Isn't it like 6 months?
It's 6 months. Why? Because again, when are these going to be susceptible? These are going to be susceptible when they're dividing. Okay? And so it doesn't penetrate well. You got to wait for them to divide. Yeah. And this is one of the reasons why we have so much drug-resistant TB, is because who's going to be sick on antibiotics for 6 months? They just don't do it. Okay. Wow. I have, it's like 4 minutes to 11. Is that right? Is that what you got? So we got 4 minutes, right? 3, okay. We're gonna do plasma membrane. You should remember the fluid mosaic model. Remember, these are lipids that are fluid, and then you've got proteins inserted, and there's all different kinds of proteins, peripheral proteins, transmembrane proteins, integral proteins, porins, all sorts of things like that that are in the membrane, fluid mosaics, so those proteins can move around, make a mosaic. Okay. Eukaryotics have what? Eukaryotic cells have what do they have? Yeah. Okay. Interesting.
Is it cereal?
What?
Cereal?
I can't hear you.
Cereal? Is that what they're called?
Steroids. They have, yeah, they're sterols. Yeah. Because sterols are from the lipid family and they thicken the plasma membrane. Okay. So you've got proteins and it's selectively permeable. What does that mean? This is a selectively permeable membrane. What does that mean? Okay, some things can pass, others can't. What can always pass a semi-permeable membrane? Water. Always. Always. Okay? All right. I think I'll stop there because what I was going to do is do an exercise here with movement of molecules, but I think we'll stop here. Okay? Cool.